All right, welcome to your flipped lesson on probability and heredity. If you're following along in your textbook with your notes, lesson 2 starts on page 80. So let's get started. We're going to start by talking about probability. I like to use a coin toss as an example of probability, something you're most probably familiar with, and we're going to do a little mini lab on this tomorrow. So each time you toss a coin, there are two possible ways that it can land. If we're tossing a quarter, it can either land heads up or tails up. So probability describes, it's a number that describes how likely an event will occur. So when we're talking about the flipped coin, in probabilities in math terms, so in mathematical terms, you can say that when I toss a coin, the probability that that coin will land heads up is 1 in 2. There are two sides to the coin. When I flip it, there's a 1 in 2 chance it's going to land on heads. There's also a 1 in 2 chance that it's going to land on tails. And so when we're talking about probabilities, we talk about them as being expressed as either a fraction or a percent. So when we're saying we're tossing a coin, there's a 1 in 2 chance that it's going to land on heads and a 1 in 2 chance it's going to land on tails. We would say that probability is 1/2 or 50%. And I know we talked about probabilities and probably didn't term it this way, but when we were talking about Mendel's experiments, we were saying that in his second generation, in the F2 generation, the offspring came out with a 3-to-1 ratio dominant to recessive traits. So 3 to 1, you could say 3/4, so 3 out of 4 of them were dominant, or 75%. The important thing to note with probability is that the law of probability predicts what is likely to occur and not actually what will occur. Okay? So if you toss that coin 20 times, if you say it's a 1 in 2 chance, you may expect it to land heads up 10 times and tails up 10 times. But in fact, you may actually get 11 heads and 9 tails, or 8 heads and 12 tails. The more tosses you make, the closer your actual results will be. But you cannot guarantee that every time you flip it, it's going to be 10 times heads and 10 times tails. Okay, it's likely, but not actually will. And this is important when we're talking about probabilities in genetics. Okay, sorry, I added an extra slide there. So we're going to talk about Punnett squares now, and we did a few Punnett squares before Christmas break, but a Punnett square is a way to sort of use or sort of visualize some of these probabilities. So a Punnett square shows all of the possible ways alleles can combine in a genetic cross. And if you remember from before break, we talked a lot about alleles in Mendel's experiments. An allele is just the kind of different forms of a gene. And we said that an organism's traits are controlled by alleles that it inherits from its parents. And when we talked about Mendel, we said that some alleles were dominant, and we represented those with a capital letter, and some of them were recessive, and we represented those with a lowercase letter. So we can use a Punnett square to help predict the probability that a trait will appear in the offspring. Now, again, it's not a guarantee, but we can help better understand what dominant and recessive traits, how often they might appear in the offspring. Okay. So let's draw a couple of Punnett squares. I'm not going to do all of these. You have some in your notes that you can follow along with. But if we're going to draw a Punnett square to help us chart out all the possible ways alleles can combine in a genetic cross, the first thing you're going to want to do, sorry about that, there we go, is to draw your square. Okay. And once you have this square drawn, the general rule of thumb is that the male's parents are written along the top and the females are along the side. And I'm going to start with, we'll do this cross first, okay? And in this cross, we have a dominant and recessive trait and 2 dominant traits. And we're going to talk about what these mean, but these are our alleles. We have uppercase R and lowercase r, and we're crossing that with uppercase R and uppercase R. So we're going to write our uppercase R and lowercase r here, and our uppercase R and uppercase R here. Okay. And how we do Punnett squares is we take these two here, R and capital R, and they go in this first box. And then we take the capital R and the lowercase r, and we put it in this box here. And then we take this capital R and this R, and we put it here. And then we take this capital R and this lowercase r, and we put it here. Okay. Wow, try to follow that. It's way more confusing than it should. So what we have here is we had two parents, and we wanted to predict the likelihood that traits would be expressed in the offspring. And we talked about with Mendel that dominant traits are expressed over recessive ones. And so when we look at these possible offspring here, we could say, even before we know much about them, that there's a 100% possibility that the offspring will express the dominant trait. So let's try one more. I'll try to avoid all of the crazy lines. Okay, so let's do this one here. So we have capital D and lowercase d, and then lowercase d and lowercase d. Okay. And so in this first box, we would put capital D, lowercase d. Always put the capital first, and we'll go over that tomorrow. In this box, we would have lowercase d, lowercase d. In this box, we would have capital D, lowercase d, and in this box, we would have lowercase d, lowercase d. Okay. Now, if we wanted to know what the probability was that the dominant trait would be expressed in this cross, we would say 50%, because 50% of the offspring have the dominant trait. Okay. And then, so 50% would be DD, and then 50% would be lowercase d, which would be recessive. So this would be dominant, and this would be recessive. Okay. So you have a couple of examples to work through Punnett squares on your notes. Take a few minutes to do those, and then we'll start kind of figuring out what all of these letters mean. So let's to this right here, genotype and phenotype. So I'm going to start over here with phenotype. Phenotype is when you look at an organism, it's the physical traits, or you could say, or visible traits. Okay. So without even knowing my genetics, or whether or not my genes are dominant or recessive, just by looking at me, you could say, I have brown hair, I have green eyes, I have detached earlobes, I have a hitchhiker thumb, I have a widow's peak. Those are all things that you can see. Now there are genes that code for all of those. I inherited those traits. You can't see the genes, but you can physically see the characteristics. That's what a phenotype is. Okay. So in Mendel's crosses, if you're looking at his offspring in the second generation, his F2 generation, you would say 3 out of 4 plants are tall. Tall is the phenotype. 1 plant out of 4 is short. Short is the phenotype. You can see it. It's a visible trait. There are genes that control those traits. You cannot see the genes, but you can see the physical traits. So that would be phenotype. Pretty easy. If you can see it, it's genetic. And we'll talk about, like, if my hair were purple, of course, that's not genetic, but just the physical traits that you can see are the phenotypes. So as opposed to what we call the genotype, and the genotype is the genetic makeup. This is the alleles. Okay. So if we were talking about if dimples were a recessive trait, we would say, and we just were going to represent that by D. Okay. That would be the genotype, and the phenotype would be dimples. The genotype, if no dimples is dominant, capital D, lowercase d would be your genotype, your phenotype would be no dimples. If we're talking about left and right-handedness, so whether or not you are left-handed or right-handed, if right-handed is dominant, this would be right-handed. This would be your genotype. This would be your phenotype. Lowercase l, then you would be left-handed. Okay. So the combination of your alleles is your genotype. How that is expressed physically is your phenotype. So I want to add something to this, and I hope that it works. Okay, so there's a couple of other vocab words when we're talking about genotype. Oops. Sorry about that. It would be good to be able to erase. Interestingly enough, my thing won't erase. Okay, so when we're talking about genotype, we can press on. Okay. We have two different types. Okay. We can have something that is called homozygous. Okay. Homozygous, and that is when the alleles are the same. Okay. Meaning homozygous would be for dimples, lowercase d. Both of these letters are the same. This would be homozygous recessive. Okay. As opposed to capital D. These are both the same alleles. This would be homozygous dominant. Okay. The other term that we're going to talk about is heterozygous. Okay. Heterozygous is when the alleles are not the same. So capital D, lowercase d, that is heterozygous. Okay. And that is always dominant expressed. So you don't have to say heterozygous dominant or heterozygous recessive. If there is a dominant trait, that is the trait that it's going to be expressed. So I'm going to stop there because this is going to be where we begin tomorrow and what some of our lab activities tomorrow are going to be focused on. So take a few minutes to review your text, review your notes. We do have a brief lecture quiz tomorrow. And good to see you today. Look forward to seeing you tomorrow.